27th November 1895. Alfred Nobel, an inventor, entrepreneur, scientist and businessman, signed his last will and testament, giving the largest share of his fortune to a series of prizes in physics, chemistry, physiology, medicine, literature and peace, the Nobel Prizes. Who can nominate the Nobel Prize? Each year, thousands of members of academics, university professors, scientists, previous Nobel Prize laureates, and members of parliamentary assemblies and others are asked to submit candidates for the Nobel Prizes for the coming year. These nominators are chosen in such a way that as many as countries and universities as possible are represented over time. After receiving all the nominations, the Nobel Prize committees of the 4 prize awarding institutions are responsible for the selection of the candidate. Who selects the Nobel Prize laureates? In his last will and testament, Alfred Nobel specifically designated the institutions responsible for the prizes he wished to be established. The Royal Swedish Academy of Sciences for the Nobel Prize in Physics and Chemistry, Karolinska Institute for the Nobel Prize in Physiology or Medicine, the Swedish Academy for the Nobel Prize in Literature, and a committee of 5 persons to be elected by the Norwegian Parliament for the Nobel Peace Prize. In this video, I will be focusing on the Nobel Prize 2024 winners in Physiology or Medicine, which is jointly awarded to Victor Ambros and Gary Ruvkun for the discovery of microRNA and its role in post-transcriptional gene regulation. Dr. Ambros is a professor of natural science at the University of Massachusetts Chan Medical School. Dr. Ruvkun is a professor of genetics at Harvard Medical School and a researcher at Massachusetts General Hospital. So, what did they do to win the Nobel Prize? And why was this revolutionary? Before their discovery, scientists thought they understood the central role of genes. DNA makes RNA and RNA makes proteins. But Ambros and Ruvkun showed that there was a new layer of control at the play, microRNAs. Let's break down their discovery. Every cell contains the same chromosomes. So every cell contains exactly the same set of genes and exactly the same set of instructions. Yet, different cell types such as muscles and nerve cells have very distinct characteristics. The electrical impulses of nerve cells are different from the rhythmic beating of heart cells. The metabolic powerhouse, that is the liver cell, is distinct to a kidney cell which filters the urea out of the blood. The light sensing abilities of the cells in retina are different in the skill set to white blood cells that produce antibodies to fight the infection. So much variety can arise from the same starting material. How do these differences arise? The answer lies in the gene regulation, which allows each cell to select only the relevant instructions. This ensures that only the correct set of genes are active in each cell type. These scientists were the first to discover microRNAs and how they exerted control on how genes are expressed differently in different tissues. They discovered microRNA, a new class of tiny RNA molecules that play a crucial role in the gene regulation. Their groundbreaking discovery revealed a completely new principle of gene regulation that turned out to be essential for multicellular organisms, including humans. It is now known that human genome codes for over 1,000 microRNAs. Let's see how they discovered this. The starting of their journey began in the early 1990s. Victor Ambros was studying a tiny worm called Caenorhabditis elegans, which is a model organism commonly used in genetics, to understand how its genes control the development, specifically how it grows from an embryo into an adult. Scientists already knew that genes in the organisms like humans and worms provide instructions to growth and function. But in Ambros' lab, something strange caught their attention. They found a gene called LIN4, which seemed important for the worm's development, but they couldn't figure out how it worked. This gene didn't seem to make proteins, which was odd because most of the genes did. So what was it doing? The key discovery happened in 1993. In 1993, after much investigation, Ambros, his team made a surprising discovery. Instead of making a protein, LIN4 produced a small RNA molecule, what we now call a microRNA. This small RNA wasn't a messenger RNA, the kind that usually helps make protein. Instead, it attached to other mRNA molecules and controlled their ability to make proteins. This was completely a new way of understanding how the genes could be regulated. Instead of simply following the traditional process of DNA, RNA, protein, this microRNA was regulating the process, telling other RNAs when to stop or slow down. Gary Ruvkun's contribution, which is in 2000. Around the same time, Gary Ruvkun, who was also studying this worm, discovered something even more astonishing. He identified another gene called LET7 that worked in a similar way. The gene also produced a microRNA that regulated multiple genes. Even more important, Ruvkun found that the LET7 wasn't just present in the worms. It existed in many species, including the humans. This suggested that microRNAs weren't just a weird thing found in worms, but something fundamental to all the life, and how they pieced it together. Both Ambros and Ruvkun realized they were into something very huge. These small RNA molecules were not just the rare occurrences, but a completely new layer of gene regulation, working across many species. Over the years, their research helped show that hundreds of microRNAs exist in humans, controlling about a third of our genes. The discovery of microRNAs wasn't an accident, but rather a result of persistent, detailed research into gene function. Here is how they approached it. So, from the initial observations in the early 90s to the realization of the broader significance in the 2000s, it took nearly a decade of research and experimentation to fully uncover the role of microRNAs. Now, the important question: how is this discovery going to impact our life? New understanding of how the cells work. MicroRNAs help regulate which genes in our body are turned on or off, acting like tiny switches. Knowing how these switches work gives scientists a deeper understanding of how our cells grow, divide, and function. This knowledge is critical for many areas of biology and medicine, including understanding disease and how our bodies maintain the balance. Fighting disease like cancer. One of the most exciting impacts is in the fight against cancer. Many cancers happen when the balance in our cells goes wrong, leading to the uncontrolled growth. Scientists have found that microRNAs play a very big role in controlling this balance. Some microRNAs stop cancer cells from growing, while others help them spread. By targeting these mRNAs, researchers could develop a new cancer therapies that slow down or even stop the disease. Number 3, better diagnosis and treatment. MicroRNAs could revolutionize personalized medicine. Since they act like indicators of what's happening inside the cells, they could be used as biomarkers for diagnosing the diseases early. For example, doctors might use miRNA levels to detect cancer or heart disease before the symptoms even appear. This would allow for earlier and more accurate diagnosis, giving the patients a better chance of successful treatment. 5. More precise medicines. Because miRNAs regulate gene expression so precisely, they could help create treatments that target specific cells or tissues in the body, reducing the side effects. For example, a future cancer drug could use miRNA technology to attack only the cancer cells while leaving the healthy cells alone, which could mean fewer side effects for the patients. These people have literally changed the world through science, and what's amazing is that it reminds us of the power of human potential. Thanks for sticking around. If you enjoyed this breakdown of the Nobel Prize, don't forget to like, share, and subscribe for more exciting contents.